So I will be talking about a project that I've been working on for about the past year of building a brain-based biomarker of whole body aging and using it to detect risk for cognitive decline. So the simple motivation for this project is that some people age more quickly than others. So to exemplify this, these are some composite photographs of people who are 45 years old. So everybody whose photograph was included is the exact same age, but you can see that the people on the left appear to be much younger than the people on the right. And this is backed up by lots of data suggesting that indeed people tend to age at different rates, which has prompted a search for accurate measurements of aging so that before the onset of disease we could identify who's at most risk of decline and who might benefit most from preventative interventions. So the question then becomes, how do you measure aging? A common way of doing this is known as age deviation. And in the realm of epigenetics, this is called the first generation of epigenetic clocks. And in the realm of brain imaging, this is known as the brain age gap. Essentially what you do is you take a large set of data from people aged 20 to 100, and then you use an algorithm to predict their age according to their DNA methylation or their brain structure. Then the difference between the predicted and the actual is thought to be evidence of accelerated or decelerated aging. There's a couple of problems with this approach, though. The first is that chronological age is confounded with lots of things that are not aging. So, for example, the year that you were born is related to how much leaded gasoline you were exposed to, whether you were exposed to COVID-19, and that sort of thing. It's also impossible to distinguish how much that deviation between the predicted and the observed is due to true biological signal and how much is due to random noise or model error. And in fact, in the realm of epigenetics, as these first-generation clocks became better at predicting chronological age, they became less sensitive to clinical outcomes like disease and death. And lastly, in a big sample of people from 20 to 100, the people who are at the very oldest ages must have been among the healthiest so that they were able to survive that long to reach those very old ages, and the people who age most quickly would not survive to be ever that old. So to get around some of these problems, our group has taken a longitudinal approach to measuring aging in a birth cohort of people followed from birth until age 45 years, and this cohort is known as the Dunedin study. In this study, it's allowed us to measure these 19 biomarkers that you see here in this list repeatedly at age 26, 32, 38, and age 45. And then we can derive an individualized slope of change for each person for each of these biomarkers. So we know how fast each person's BMI or their CRP is changing over this 20-year period. And then we can collapse those rates of change into one overall slope that we call the pace of aging. So this pace of aging measurement is very useful within our cohort and is very informative. And we also developed an epigenetic tool to estimate it, which is known as Dunedin pace. And Dunedin pace has been widely adopted now in many different cohorts and studies, and is generally thought to be more effective at identifying clinical outcomes than first-generation age deviation epigenetic approaches. However, there is no such neuroimaging style metric as of yet, and the neuroimaging-based aging measures are almost all age deviation-based. So here, we're asking whether or not we can build a neuroimaging-based aging biomarker that estimates longitudinal aging. If so, we want to ask, would such a biomarker be useful in an external data set outside of our cohort in identifying risk for cognitive decline? And would this biomarker be different from age deviation measures that have already been developed by other groups? So we begin with question 1 and use a sample of 860 neuroimaging structural MRI scans from our study members when they were 45 years old. And we use those scans to build an elastic net regression model that estimates their pace of aging at age 45. And we call the resulting scores Dunedin-Packney, which stands for pace of aging calculated from neuroimaging. So we see that we're able to predict the pace of aging within our study members with fairly good accuracy. The in-sample correlation is 0.6, and the cross-validated accuracy is 0.42. So that's okay. It's not perfect. But importantly, what we see within our study is that the Dunedin-Packney scores capture the magnitude of associations with all these aging phenotypes at a similar magnitude as the original pace of aging does. So we see across measures of physical functioning, cognition, and facial aging, which is that metric I showed you at the very beginning. Dunedin-Packney tends to find similar effect sizes as the original pace of aging. So next, we want to know, does this biomarker, which we've trained in our sample in New Zealand, generalize and tell us something useful about people in different data sets? So we took our elastic net regression model, and then we applied it in the ADNI data set and UK Biobank data set. ADNI is a large neuroimaging data set of people enriched for people with mild cognitive impairment and dementia, and the UK Biobank is a large neuroimaging data set of basically healthy adults. So first, we tested whether or not Dunedin-Packney scores were just cross-sectionally associated with cognition. So this is a forest plot that shows the absolute effect sizes standardized across different tests. And we see that across both studies, faster Dunedin-Packney scores are associated cross-sectionally with poorer cognition. And across different domains that include mental status, memory, processing speed, executive functioning. We also see that people who meet criteria for clinical cognitive impairment in the ADNI study, on average, have higher Dunedin-Packney scores compared to people who are cognitively normal within the ADNI study. So we find these cross-sectional associations, but we also want to test whether or not Dunedin-Packney can tell us something interesting about future risk. So we took a set of ADNI participants who are cognitively normal at baseline, approximately 600 people, 112 of them converted to either MCI or dementia during the follow-up. And we see that people who had faster Dunedin-Packney scores at the time of their first scan were more likely to cognitively decline during the follow-up period. We also repeated this analysis with people who had mild cognitive impairment already at baseline and saw that people with faster scores at baseline were more likely to convert to dementia during the follow-up. So now in the last part of the talk, I will I'll answer this question number 3 of how this Dunedin-Packney score is related to a first-generation brain age gap score in ADNI and UK Biobank. So we see that the correlation between Dunedin-Packney and brain age gap, shown here in these two scatter plots, is not that strong. So in ADNI, it's about 0.17, and in UK Biobank, it's about 0.3. We also see when we conduct all of these phenotypic associations in ADNI and in UK Biobank, including a bunch that I have not talked about, we see that in general, Dunedin-Packney effect sizes are slightly bigger or roughly the same as the effect sizes for the brain age gap. In this plot, again, I'm showing absolute standardized effect sizes, and Dunedin-Packney is shown in turquoise while brain age gap is shown in pink. Now, because they're poorly correlated, we wanted to ask whether when you control for one biomarker, what happens to the effect size for the other? Are they soaking up different amounts of variance? So in this scatter plot, I've now overlaid lighter shade effect sizes for the effects when you're controlling for the other biomarker. So you see, in all cases, the effects are slightly weakened, but not by very much. So our takeaway from this is that our Dunedin-Packney score and the brain age gap score are soaking up different variants, are differentially measuring aging that is relevant for these various health outcomes. So to conclude, we believe that structural neuroimaging data can be used to directly estimate longitudinal aging. This is instantiated by Dunedin-Packney, which we find is associated with poor cognition, cognitive decline, and risk for dementia. And we see that Dunedin-Packney is distinct from the brain age gap, which is an age deviation measure based off of brain structure. So I'd like to acknowledge everybody in my group at Duke in the Moffitt, Caspi, and Hariri labs, everybody, all of our collaborators at the University of Otago, and our collaborator Max Elliott at Harvard, and then also point out that we have a preprint on this on bioRxiv, which you should check out if you're interested to learn more. Thank you very much.
For the Packney score, based on how you developed it, I'm curious your thoughts about the age groups, the age bins in which it's going to have the best validity, and what that validity might look like, in perhaps very young people, but also older people with neuropathologies.
Yeah, so that's a great question. I went too far. We see in both ADNI and UK Biobank that there's a positive correlation between age and Dunedin-Packney, which is exactly what we would expect for an aging biomarker, right? That as people get chronologically older, also the rate at which they age increases. So there's this exponential relationship. So the fact that we see this correlation makes us heartened, and we think it's a validation of the measure. At the same time, it's very possible that our score may have less relevance for detecting AD pathology, given that probably a very small proportion or nobody in the training set would have had substantial AD pathology at the time of their scans. Cool. Thank you.
Much.